It's absolutely no question that cancer is on the rise. Soon, everyone is on track to personally know someone who's combated the disease in their lifetime. In 2021, it was the second biggest killer in the United States, and actually the whole world in 2023, only falling behind heart disease. And to make matters even scarier is that cancer has been killing species since the age of the dinosaurs. Every time period since has seen it being a massive fight. But it bodes the question, why is it still here? Why haven't we managed to kill it yet? It turns out that even though we've come a long way, unfortunately a lot of chemotherapy, our current favoured treatment, is rather unsuccessful. Nevertheless, it seems we have so many new methods which may be replacing chemotherapy in the near future, some of which are so innovative you'll likely have never heard of them, and all we can do is hope they're used alongside it. Here is how we now kill cancer, with the hope of finally defeating it forever, before it continues killing more people. Let's start by addressing what cancer actually is. Cancer is rapid uncontrolled cell division. Oftentimes it's caused by changes to the DNA within cells. Specifically, our DNA contains two types of genes on it, of which both are linked to cancer. One of the two is proto-oncogenes. Healthy ones stimulate normal cell division, producing proteins to make cells divide. Conversely, we have tumour suppressor genes. When these function normally, they slow cell division by again producing proteins or just destroying the cell completely. If a mutation occurs, two things may happen. If it's the proto-oncogene that's mutated, it'll become overactive and cells will divide uncontrollably, essentially causing cancer. And this new gene is called an oncogene. If the tumour suppressor gene becomes mutated though, it will instead become inactive. Should this happen, it will not stop standard cell division from the proto-oncogene, and uncontrolled cell division will again occur. Over time, these cells will clump together, forming a tumour. Now, our main treatment we use in the present is chemotherapy. Chemotherapy itself is an anti-cancer drug that seeks to interfere with this uncontrolled cell cycle. Unfortunately, it does tend to affect healthy cells too, but let's focus on the good for now. When these cells are rapidly dividing, as seen in cancer, chemotherapy drugs can directly target this DNA, destroying the new cells and potentially the existing ones with this faulty mutation. But if we take this a little further, it's been found that specifically the p53 gene is perhaps more crucial in the cellular response to DNA damage. At this point, the majority of chemotherapy drugs tend to focus on targeting this precise pathway with the hopes of destroying the cancerous tissue. Without this gene, it becomes a lot harder for cells to rapidly divide, as seen in cancer. Yet the primary problem is, as said, this doesn't seem to be working in all cases, and it's time we look at some other theories in how to more successfully kill cancer. One of the primary reasons we don't really use chemotherapy, despite its decent efficacy, is the side effects. People undergoing treatment become incredibly fatigued, unwell, lose their hair, and can even get anemia. It's also the same reason we don't really use X-rays. But what if I told you we can in some instances kill cancer without all these side effects? This method is called proton therapy and it's relatively new. It's basically a beam of proton particles that stop at the tumour, unlike X-rays that penetrate it. This in general has healthy tissue nearby remain relatively intact, and in a study from JAMA Oncology, whilst patients tended to have similar lifespans and a range of cancers, using both types of treatment revealed those undergoing proton therapy had significantly less symptoms. This is 12% in the proton therapy group and a whopping 28% in the traditional radiation group. With it, people could generally continue their days and lives, some even vacuuming, painting, working. It didn't really inhibit them at all. Yet this study and proton therapy as a whole still isn't perfect. The study mainly saw older, more ill patients receive the special therapy. Perhaps their negative symptoms were already present. Plus, proton therapy is also insanely expensive. Not to mention there's actually minimal research if it does work without such side effects. So for now, let's consider some other options. Perhaps another uniquely strange idea is the use of bacteriophages. These are little viruses that infect and replicate within bacteria. They're honestly amazing little wonders, and their use has already been cemented in the treating of superbugs. But can they kill cancer? Well, a lot of research has explored this idea, and in fact it was found that there are potential properties of bacteriophages that have been found to directly kill tumours. Such phages are specifically designed to infect the antigens of cancer cells which can trigger their death. Specifically, their ligands can be genetically engineered to recognise and bind to selected cancer cells. So it appears that healthy tissue in general will be spared. There's even been links to these bacteriophages enhancing the immune system, ultimately aiding the fight, and also altering the microbiome. This refers to the composition of bacteria within the tumour's own microenvironment. Some researchers found that phages can actually alter this, affecting how the tumour responds to other treatments, potentially allowing for the usage of both. Here's the problem though, most of these humours are heterogeneous, they're not the same. Manufacturing specific bacteriophages is a huge challenge, and even so, they may end up simply triggering the body to kill them before they actually kill the cancer, being less effective than the chemotherapy altogether. And even so, we can develop resistance to phages. If we use them all the time and rely on them for so long, that's an entirely new problem which can result in death. Another kind of virus though is oncolytic ones. Instead of replicating in bacteria, they replicate in mammalian cells, particularly cancerous ones. The literature has posed that it's very promising and has many positive characteristics. For example, they're naturally designed to lyse in the tumour cell, leading to the release of new viruses when they self-destruct. One such virus has been used so far, and it's called TVEC, which is basically just herpes simplex modified. In fact, several viruses are currently being trialled, including measles, adenovirus, reovirus, and Newcastle disease virus. And similarly, it's good with other therapies. In fact, in 2019, Harrington found that when combined with immunostimulatory cytokines, 1 in 6 patients responded and 8 out of 9 were cured at least 5 years. Now, keep in mind this is new, so even though this doesn't sound amazing, it's still really good for a starting point. However, with this option, we're almost back where we started. The side effects can consist of the immune system turning onto healthy cells and severe infection, as well having heavy fatigue and nausea. As we've seen though, it's still not completely effective, and we are in need of greater mechanisms to finally kill cancer. But what if we go back directly to where it all started, the faulty genes themselves? Gene therapy can see this through. This is a process where someone's genes are altered in what is oftentimes a controversial process. See one of my latest videos for reference. So how does this one work? Well, if you remember the proto-oncogenes and tumour suppressor genes we touched on earlier, we can actually afflict cancer's progression through gene therapy. We can inhibit proto-oncogenes through introducing suicide genes, coding for enzymes capable of converting non-toxic substances into substances that will kill the cancer, or to even have siRNA delivered to cancer cells, inhibiting their expression. Conversely, we can introduce functional copies of tumour suppressor genes or enhance their activity. This is done through CRISPR editing. When used clinically, there was in general a 90 to 96% response rate, however only around 35% of people responded completely. So even though it can clearly kill cancer, it's still not foolproof, and a lot of people still need other treatment. Nowadays, we can harness most things in our fights, and it seems in this method of killing cancer, we've done so with the temperature. Both cryoablation and hypothermia have seen promising results, with the former referring to a thin needle, known as a cryoprobe, inserted through the skin into the cancer. Gas is eventually pumped inside, which freezes it, and the tissue simply thaws and dies. Hypothermia, on the other hand, burns them, both keeping healthy tissue intact. When hypothermia treatment was added to simple chemotherapy, the efficacy rose from 35% all the way to 80%. Cryoablation, on the other hand, saw, in at least kidney cancer, a 100% survival rate at 5 years. Yes, everyone survived, which is honestly groundbreaking. But even though it is, this hasn't been replicated in every instance of cancer. There's been trials with bone, breast, cervical and eye cancer that don't quite have 100% efficacy with the results displayed on screen, but it is a good starting point. Knowing that we can combine these therapies is certainly something. Perhaps the last method, and potentially the most extreme in my opinion, is mitochondrial targeting. Your mitochondria are an organelle in your cells which are responsible for energy conversion, turning oxygen and glucose into ATP. I'm sure you remember from biology class that energy is never lost or destroyed. The literature has suggested that we can use this to our advantage. If you give mitochondrial antioxidants such as MitoQ or MitoTempo, they'll eventually accumulate in the mitochondria and they'll cause the cell to die because it won't have any energy to respire. As of now, there's not a lot of research on this, but initial trials do seem actually quite promising. The only risk is if we accidentally infect cells that are not cancerous and the patient would likely end up dying. So it's a little bit of a toss-up. So there we have it. Obviously, we have many prevention strategies too, some equally peculiar, including alkaline diets, coffee enemas, and positive thinking. Yes, I know what you're thinking. But it is important we remember that even though at times our fight against cancer looks bleak, we have so many new, evolving methods that are proving effective. And who knows, maybe in 20 years we'll have even more methods and we'll finally be able to kill cancer forever, something our ancestors could never do. Thank you so much for watching. I appreciate every single one of you. Be sure to like and subscribe if you enjoyed. It's free and it really helps me out. See you next time.